Pablo, nice to see you again.
Hello, Marion. How you doing?
I'm great. I'm so excited to be talking to you again. It was fantastic to talk to you in Spanish last time, so we're gonna do another little episode about you and your experience, this time in English, for the benefit of everyone.
Thank you very much, and I hope you like the Spanish version of our interview.
Yes, fantastic. So, Pablo, please tell us about your training. How did you get into mucosal immunology? Where did you train?
Okay, so I'm trained as a biochemist here in Argentina, and then I moved to the US where I applied to different universities to get into a PhD program, which I thought it would be great to highlight because it will explain a little bit of what we are doing around here. Um, After taking the GRE subject, there are these standardized exams so you can compete and you have to write why the university has to invest in you. You get into these great programs and you're going to go for a year with basic courses that are going to give you great training and the basic foundation to understand the science that you're going to do. And you're going to rotate probably in 3 labs. Um, I had the great opportunity of doing my PhD at Emory University, which I'm very grateful about. I did my PhD with John Altman. He was the first to design the T cell tetramers, if you know about them, and now he leads the NIH tetramer core there at the Emory Vaccine Center at the Yerkes Primate Center. So I had the opportunity to be exposed to great science, and I was participating every week of seminars where professors from different universities will come to Emory to talk about their research, and usually will be probably a recently published or soon to be published paper. So it's great to see the science happening right in front of your eyes. And then you can see even people that have gone into win Nobel Prizes um, that have sat with you and they have talked to you one-on-one, listening to your PhD project and giving you insights. That's amazing. It's great to see this science being personal. You can do these seminars. You can see journal clubs with people that are really experts in the field, and you can learn how to approach an article, how to understand how they did it. And on top of that, you get the exposure to the great technology around. Then you will get your project going in a thesis committee formed with great scientists, and then you will end up going to national meetings that are great. Also in America, that you have lots of people attending the American Association of Immunology. But how do you get SMI involved in this? SMI, in my opinion, is immunology 2.0. It gets to a field that is not really covered entirely by the American Association of Immunology, or I think the European Immunology Association may be in the same place. And so SMI brings this plus to a field that is not really covered in these national meetings, but also has even its own book. You have the book of Principles of Mucosal Immunology, and not many really universities are teaching this, which is a great opportunity to help spreading this knowledge by SMI. So, from my opinion, I think my training got me in my case, I was studying how CD8 T cells and the first interactions will impact in their memory differentiation. But you will get to talk to a lot of people that were doing great science and it will improve what you're doing. And I think that interaction is something that makes you grow a lot. And I think SMA provide that opportunity, and particularly in places like what you were mentioning in Australia, probably you don't get to interact with as many scientists, or here in Latin America. So I think that those opportunities for training were something that I really liked. I move on to my postdoc by going into one of these meetings of national meetings. I was able to get a fellowship at Emory to teach my research to undergraduates. So then you get to teach, and when you teach, it's like you learn again what you're doing. You can get a deeper understanding of what you're doing by explaining it to somebody else. And so that opportunity was also there in Emory. And then I got to a national meeting where I met Leo Lefrancois. He came by to my poster, and you will think, well, how is this possible? Many of the meetings will provide that opportunity. If you're a trainee and you interact with SMI, this is what is going to happen. You're going to have people that are experts in the field, and they will come and they will see what kind of science you're doing, and then you will move on. And I wanted to know more about cellular immunology. And Leo said, well, you know what? We love gut over here. No guts, no glory. So you have to work on gut. And we have this great machine. We had a brand new 2-photon microscope that somebody had to run it. And I said, well, what is the hypothesis here? And we need to make great pictures. And sometimes when science starts that way, we want great pictures, but also you have the opportunity of getting into, for example, I was able to see how gamma delta T cells form memory. This was the first time we were seeing gamma delta T cells in mice forming memory, and I was able to image them. I was able to see them with confocal microscopy and 2-photon microscopy. I was able to see their speed and how they were interacting. That was amazing and a great opportunity. And that's something you can do in your postdoc to get trained, but also with technologies that are not usually available next to you. And from then on, I got to see my first SMI meeting in Germany, which it blew my mind. If I knew God and I really like it, going to SMI and so and seeing all these experts, really, I felt at home in science. I felt that this is the place where I wanted to be, where I wanted to interact. It was impossible to see all the talks and all the posters, but this is how you feel, at least in my case, about SMI interacting. And that's how I got to know SMI. And I finished my postdoc thinking on how I'm going to move next, going back home.
So you were thinking of going back home, back to Argentina, right? So where science is done in a different way, perhaps, where facilities are different, etc. Can you tell us a little bit?
Yes. So I was telling you how we were doing the research and the training in US. Coming back to Argentina, usually you associate with professors that have already a lab, established lab, because there are not that many resources. So you enter in a career at CONICET, for example. There are different hierarchies, and you start with asistente, then adjunto, then you go to independent, and then principal. You usually join a lab that already has all the techniques established, and you're going to interact with them. And you're going to have something that I really really like to highlight here. I love going into mountains. I was a Boy Scout, a Master Scout too. And I used to go a lot of time and still going and even climb the Mount Washington in US with Leo. But going to the mountain, you always think about how approach going up this mountain. So training in your PhD is like going up this mountain. Going to your postdoc is going up this mountain. But usually going up a mountain is not easy if you don't know how to do that. Usually you will talk to somebody, a guide that has done it. And I think that's a key piece of this. This is what SMI also provides. The mentorship program is the guide that will help you get to the top of the mountain. This is what you need in science. And it's something that if you look back at some countries, not all the countries set up a guide for you. Usually if you join a faculty position in US, you will have a mentor that will take care of you. You probably will meet every month or every 2 months, but there is always a person that is looking out for your career. And I really like to point out the series that Nature Career Center has. And by no means I intend to do any publicize any other place, but in Nature Careers there are many articles that will talk about mentorship and how to do it. And one of the things that struck me the most is, as a mentor, you should get in the shoes of your mentee. And after knowing everything that you know, now in these shoes, what would you do in this step of your career? So we need mentors that think that way for you. And SMI provides a mentorship program so you can tag along with somebody that already went up the mountain. So I chose E4 Williams. He was a professor that I met at Emory University, and he helped me a lot coming back to Argentina, setting up my lab, but I also had the help of a local mentor. My local mentor was Fernando Chirdo, in La Plata. He bought one of the first reagents, but I also would like to tell about the now dean of the chemical sciences, which is Silvia Correa. Silvia Correa was a professor in my biochemist degree, but also had the luck of coming back, interacting with her, and we created one of the opportunities that was missing also here. I was talking about the seminars in US and seeing people every week. Well, over here we created the mucosal seminars. We were having once a month one article from mucosal immunology between our two groups. So we started building opportunities to train the students here for something that was missing. And that's how, interacting with E4, with Silvia, with Fernando, the idea came of creating a course that will supply all these things that are missing in Latin America. But need to step back. Fernando did his PhD in Argentina, and then he couldn't do a postdoc outside, but he did a short fellowship with Dr. Alan Mowat, who was the president, one of the ex-presidents of SMI. And so Alan Mowat proposed the idea of creating a Latin American mucosal immunology group to Fernando and Ana Caetano from Brazil. And that's how the idea of LAMIG came to be. They started recruiting great scientists that were doing mucosal immunology in Latin America in different countries, and forming, particularly led by Fernando for a lot of time, and now is moving into creating its own board, bylaws, and thinking about providing not only these training opportunities by seminar where we can interact. There is one now coming up in Bogota. But coming back to the idea, Fernando and Alan Mowat had this same view of there are things that are missing here. We need to supply. And that's how the advanced course in mucosal immunology came to be. We invited 50 students from all Latin America. SMI was a great supporter of this. E4 Williams, part of the mentorship program, helped a lot. And we invited experts. In each of the lectures, we had the expert in the world. We were lucky to be part of SMI and invite who is the one that knows the most. We tried at least, and we got most of it. And they come and they interact with you. They see your poster, they talk to you. We had journal clubs, and so you will see them how they will pull apart a paper. The same things that I was seeing in my PhD, I was seeing happening in this course. So to me, it's like, if you can dream up a course, this is my dream. It became true. And so it was journal club, it was the best lectures, it was interaction. And we are under now a project of trying to see what was the impact of ACME in your life, to all the students, to see what happened, who did you meet, how this course opened up the door for your postdoc. And if you think from the other part of the world, where you have all the training opportunities and you have this great lab, but sometimes you cannot find students, this course will provide a great cohort of well-trained students to advance your lab. So I think it's a win-win for the world, and these training opportunities we were lucky to be part of.
That's all fantastic. Such great success stories. Congratulations on being part of this. It's such a dream. So please, you are talking about this cohort and how you are seeing that SMI really is impacting the career of those students in those cohorts. What could you say was the impact of SMI in your academic career? So you went to the US, you trained with great people over there. came back. But what about SMI? Because SMI knows no border, right? This is not a national society, it's a global society. So how was this kind of distinct from, how was the impact distinct from the impact of your mentors over there in the US?
Well, I think it's by building your science here, there are not that many resources. And this is one of the ideas that Fernando had in his mind. He was really the precursor of these courses. He was seeing how difficult it was to be trained here. And so, and to access these opportunities of interacting with leaders, interacting with technology. And so I see SMI providing this huge amount of people that are experts in the field that will interact with you through these training courses, the mentorship program, the traveling technique, the technique sharing opportunity, because then things that you don't get to see around here, you can get in another lab and see and learn this new technique that is not around. For example, to me, to learn 2-photon, I had to go to Mark Miller's lab and see how it is. And it was easy. It was Connecticut to Missouri. But now we're talking Argentina to US or Europe, and it's not that easy. But technique sharing is making this possible. And SMI is making this possible, providing opportunities of training, providing opportunities of interacting, and providing funding, which I think we can still work more on that. There is these opportunities of doing chapters. I believe we would love to do some of the chapters that get junior scientists together. I still think there are opportunities, particularly in this context of the world, where coming around the first grant to start your lab is very difficult, particularly in our areas. You can probably tell us how was in Mexico. Would it help to have, for example, a small grant just to get your first paper out and get your lab? Things like that will be great coming up from SMI. How was it in your case?
Yes, thank you, Pablo, for raising this. So first, I have to say that I was the recipient of a technique sharing grant. I visited Megan Koch Lab in Seattle, and I am into the current cohort of the mentoring program. And SMI, like for me, has been the best society that in my short career in mucosal immunology, it already gave me so much, you know. Plus, it's super cheap for me because I am in Latin America, so I could enjoy the principles of mucosal immunology course. It's been really great. And then, so my next question was going to be about what you think SMI could do to further help scientists in Latin America, but you already went into this, so you have some ideas on possibly trying to get this call for young scientists to get their first paper out, helping a little bit with reagents or something like that, right?
I think SMI is, we discussed this in our other interview, that the people in charge of SMI, of the vision, because sometimes people can change, the vision of SMI has been to help this training of young scientists, which is a great strategy. I think it helps a lot. For us in Latin America, at least for ICME 2017, it was the area that grew the most in terms of scientists. It's really giving us a lot of opportunities that is showing on how much participation Latin America is having now in SMI. Brazil in particular, Dr. Ana Maria Caetano Faria has created this new institute of mucosal immunology that has congregated around 39 scientists from different countries to invest in mucosal immunology. And so I think SMI is the backbone that makes this possible. And the interactions, I have a PhD student now in San Pablo doing technique sharing, and it makes your paper to evolve and to be publishable into an international level. Also having mucosal immunology as a journal provides lots of opportunities. So I think SMI is doing a lot, and it's providing things that are not easy to come by in our places.
Absolutely, I agree. Thank you very much for articulating this so nicely. So I would like to ask now a little bit more about your research, Pablo. Can you tell us what you're working on now in terms of
Sure.
development?
So
Yeah,
my story was a little bit, it has many details of bumps in between, but my idea was always I fell in love with the gut, which may sound terrible, but there is so much to learn. Probably lots of scientists fell in love with different fields. But in my case, I decided to look into this host pathogen interaction, and, uh, and, and, and in this, it gets you a little bit into the frontier. To me, as a young scientist, it feels like you get trained in a field, but then you move to the frontier, to what is not known. And that usually takes you out of the main field. And, and in my case, it put me in between microbiology, in between nutrition and metabolomics, things that are not really taught in immunology, classical immunology, but now are getting part of it. And so I'm looking into metabolites. I'm studying one of the tryptophan metabolites, the indole 3 propionic acid, that is getting a lot of press lately. And we hope to get our story out. But how is it that impacts in the mucosa? And if you think about the tons of metabolites the microbiota creates, we have a lot to do. We have a lot to learn about these metabolites. I'm also looking into IBD, and I have this simple question, but it has not been answered yet completely. If you take the feces of a person with IBD and put it into a mouse, that gut is going to get inflamed. So there is something in that microbiota that inflames the gut of these new animal models. And so now my question is, what is the identity of the species of microbiota that causes inflammation? And that's part of another PhD student. And then I'm moving into another frontier, because it wasn't just easy to move into a frontier where nobody was, that I got into malnutrition. And part, part of the problems that you face in Latin America is this pathogen host interaction with lots of tropical diseases, for example, in Brazil or in our places. But also we have malnutrition, part of our economical situation. And particularly here in Cordoba, 1 in 2 kids are poor. This is tough for us. And 80% of the new moms that are low resources are malnourished. They are overweight or obese. And so my question was, how is it, from what we know from the microbiota and the metabolomics and the diet and how they impact the immune system, how is it that malnutrition changes microbiota? And how is it the metabolomics of these future moms are going to impact in the neonates too? So this is a new area. We have a PhD student working on that. We have a cohort of pregnant women in the first trimester in 2 institutions, in a private, in a public institution. And we are already seeing diet differences, difference in microbiota, differences in metabolomics. And my projects at this moment are really between building teams, because I have to interact with a nutritional practitioner, with physicians, with nurses. So we need to build teams to do these kind of studies, to involve human patients or participants, and to ask this question: what is it? What are the problems that we have? And I got the opportunity of being in a center that focus on solving the problems that we have here. So what are the questions that a physician sees? And one of the things that we are doing is training the player that is missing in the team. We think in our countries there is a player that is missing, which is a physician doing research. So I'm training 2 physicians to do research. It's hard because the physician is in our areas are not used to getting to research, and they're not paid to do research, and most of them do it for free. This is like a hobby. So it's hard to explain to a physician that works in US and Europe that our physicians really don't get paid to do research, and it's hard to do these kind of teams. And so within this building, my projects, I focus on opening up new doors. So in the questions, for example, with the metabolite, what is it doing in the gut? I'm thinking on preclinical observations because I'm thinking this metabolite could be used in humans and be a solution as a therapy. If I ask the question, what are the microbiota species that are causing inflammation, I'm thinking we can target those. We can do phagosome therapy to get rid of some of the species that are causing this inflammation. In terms of malnutrition, we can even interact with people developing food, like the group has done in Bangladesh from Jeff Gordon in the Washington University. They have seen that you need to work on providing the right type of food to your microbiota to mature and get it from malnutrition into healthy. And so those interactions from our training in immunology being applied to solve problems in your own place. So those are the research that I'm most proud of, because it's trying to solve questions from the immunology point of view, but then solving problems in your own society. And then I participated in two more type of research, which could be considered helicopter research. And this is something we need to talk about, because in our countries, we don't have resources usually, but we do have problems and participants that could be part of groups that are being funded in Europe or in US. And it could be thought that somebody just wants your samples and that's it. And what do you gain as a scientist? And so the idea of interacting, building your capacities with the opportunities of funding that far exceed what you can get in your own countries, like in Mexico and Argentina. You can get, for example, an Horizon 2020, and I was able to apply my knowledge in microscopy into asking the questions, can we predict liver cancer, for example? And we were able to acquire a new microscope here, which is an Akoya Biosciences. Sorry, not trying to publicize, but to do multiplex and to ask the questions, what are the immune cells that are infiltrating the liver before cancer starts? Or we are part of the Jaguar initiative led by Sanger Institute, in which we participate with Alejandra Medina from Mexico and many other bioinformatics from all Latin America, with the question of what is the diversity of the immune system in our countries, in Latin America. So we have in Uruguay, in Chile, Argentina, Peru, Colombia, Brazil, and Mexico. And we are taking samples from them, and we are doing single cell RNA-seq, doing site-seq, single cell attack-seq, and many other, I think in EQTL, always depending on how much money you can get. But this type of research gets you funding and access to technology that is not here, but you can participate and build your own capacities. And it is always the first step. As a young scientist, the first experiments are gonna probably create the new lines. Like we were talking before, this is like having your own garden, seeding your garden, and then seeing the plants going up and picking up the plants. Some will grow, some others probably will stop there. But that's the science that I'm doing over here.
Thank you. That is so cool, Pablo. Really, congratulations on reaching all those successes. Your seeds have definitely grown, most of them, I think, from what we heard. Thank you so much for sharing your time with us and your experience. Looking forward to see you at the next ICMI conference.
Thank you so much for this opportunity to SMI, for your presence, Marion. It has been really helpful, and I'm very happy that you can share things that we have in similar
Absolutely.
experiences.
Absolutely.
Okay.
Thank you.
Thank you so much.